Okay, let's roll. So we last left off, we introduced to pretty much all the players in the mechanism. This is the first step of the mechanism, if you recall. This is the first piece. We got introduced to our last player, that's the clamp loader, the squid. And the big squid in the middle is basically attached to just about everything, right? So it's essentially attached to both helices in a couple of places, primase. It's got its hands on the two polymerases as well. And it's got an entire domain that's designed to essentially grab onto the clamp and load the clamp. So this is the first step. So here you can see DNA polymerase, which is already started. So it's already kind of launched off of its little primer here. And essentially it's basically moving through the open template, removing the single-stranded binding proteins and replacing that with DNA. And then it'll bump up against this last primer here, and then it will just drop off. So it's not going to seal it all the way because that's not what polymerases do, right? Polymerases don't do the backbone sealing thing. It's a totally different reaction. All they do is dehydration synthesis. That means all they do is add stuff to it, right? So they can only add, they can't finish, okay? So what that means is you have all these like little nicks all over the place that need to be fixed. Talk about that here in a little bit. Notice what's happening though behind the polymerase is the helicase has already opened up new fresh template, which has already been protected by single-stranded binding protein. And you've got an active primase, which is busy laying down your primer, getting that previous segment ready. So what you have is your helicase, which is continuing to plow into the replication fork, opening up new template, primase priming the new template, and of course the DNA polymerase finishing that off and filling in the gaps as those primers are laid down. Now that's the lagging strand, right? The leading strand is easy because the leading strand basically just gets started on a primer and you just motor forward. That's it. Okay. And there's nothing that you need to do. You just keep going until you run out of template. Okay. So that's the easy part. Now in step 2, here you can see is we have the DNA polymerase, which is just about finished with its piece. It's got 2 more nucleotides to remove, at least single-stranded binding proteins to remove, and then replace that with the nucleotides. And then once it finishes, then it's got to basically re-engage. It's got to fall off and it's got to re-engage to this primer over here. Now notice what's happened. Once that primer is laid down, your primase exit, right? So that is your clamp loader, let's go, allows the primase to fall off. Remember, all the while, helicase is continuing to unwind and open up new template. So once you finish the segment, then what happens? Well, then what happens is basically you fall off, right? So here you've basically finished that one segment. So here's that primer you started off with. You've already fallen off. So DNA polymerase already dissociates at this point. But notice what happens. DNA polymerase is hung on to by the clamp loader. So the clamp loader never lets go of polymerase. Okay, so a lot of times when we look at this, we think, okay, the DNA polymerase just sort of randomly falls off, and then it kind of finds its way magically back on. That's not the case. In this case, the clamp loader is orchestrating everything. So it hangs on to that polymerase. And notice also what happens in this dissociation process is the clamp lets go. Or the clamp is the one that finds the DNA and essentially provides a binding point for the polymerase itself. So if you want to disassociate the polymerase, you undo the clamp and the polymerase will fall off. Of course, it doesn't really fall off. It's pulled off by the clamp loader. The clamp loader is always holding on to it. Okay. And so here you can see the clamp loader hanging on to DNA polymerase. The clamp has been released. DNA pol 1 is doing its thing, right? Removing the primer, gets to work immediately, and replacing that with DNA. But now you have to basically reassemble at this new prime site, right? So you have to figure out how to get your clamp there, and then you have to figure out how to get your polymerase there. Well, that's where the clamp loader comes into play. So the clamp loader will grab onto the clamp and literally clamp it onto the DNA right by the primer. And then once it does that, it takes the polymerase, which is already hanging onto, and then attaches it to the clamp. So in both cases, the clamp loader is basically purposely taking both pieces clamp and polymerase, and then assembling them at the new priming position. And that's what you see in step 4, this one. So here you can see the clamp loader is grabbed onto the clamp. It's assembling that clamp at the primed position, and it's bringing the polymerase in and basically connecting it to the clamp. Once that happens, then the clamp can basically just motor along and slide that polymerase along the open template and create that new DNA. However, notice at this point, the clamp loader never lets go of helicase, never lets go of the leading strand. That one's always going. But as you start to open up new template, your primase has to come back and reprime. At this particular point, once this is assembling further upstream to do this new segment, notice DNA pol 1 has finished. So it's already removed the primer, and what you have left is just a little nick, and that's the job of DNA ligase, who'll come in there and just basically seal up that nick. Once you do that, then you're done. You've got your DNA molecule. Yep.
The protein on the far left,
This is the gyrase. So that's that sort of topoisomerase, that smoothase, right? That one that kind of goes out and smooths all that coiling out ahead of time. So then once you finish with this and you've got it assembled, notice you've got your polymerase complex back in action, making that one segment of DNA. But notice as that's happening, helicase is opening up new template, and then in comes the primase again to lay down that new primer. And so you continue over and over and over until you run out of DNA to prime. And that's the basic mechanism. So this is basically the cast of characters and what they do. So if you can take a look at this table and you can explain in good detail what each one of these guys is doing, then you've got a pretty good start on the replication chapter. Because the story of what these guys are doing is essentially the story of what's happening with leading and lagging strand. Okay, so that was all prokaryotic, right? Now, what about eukaryotic? Is the story the same in eukaryotes? Well, the answer is yes and no, right? The basics are similar. For instance, the enzymology. The enzymes do similar sorts of things. However, there are some details that are different. That's always true with eukaryotes. For instance, we have a lot more DNA in eukaryotes. We have actually separate chromosomes, not just one circle. So it's very easy. Just grab onto that one circle and away we go. So we have all these chromosomes floating all over the place. So we kind of have a little bit of an Easter egg hunt on our hands. So we have to kind of coordinate all of this together. So that's a little bit more of a challenge. We also have linear chromosomes, not circular chromosomes. Why is that a big deal? Because when you're linear, that means you have ends, right? And ends are not necessarily a bad thing, but in this case, they're somewhat problematic. So let's kind of highlight a couple of the major differences. Origins. Let's start off with this one. We already know that bacteria have an origin of replication. They also have a termination site, don't they? Eukaryotes don't have a termination site. So that's one that's off the list. The other one is they don't just have an origin, they have lots of origins. And know that, but they're not organized the same way that prokaryotes are because they're linear. They don't have just a single one. They have multiples, which means they have, instead of replication force, we think of eukaryotes as having replication bubbles. As a matter of fact, when we draw the replication fork, that's what we're doing, is drawing half of the bubble. Essentially is what we're doing. So in eukaryotes, we see these replication bubbles and lots of them. Each one of those represents an origin. Which makes a lot of sense, right? I mean, you got a lot more DNA, so you got a lot of work to do. Not only that, but you got a lot of chromosomes, so you're gonna have to wrap your arms around all this. So does it make a lot of sense to just have one origin and then hope that you can get everything done? No, it makes a lot more sense to attack it on multiple ends, right? So basically create as many origins as possible and get the job done, right? Because you got a lot of work to do. And so that's also the case. The other big thing also with origins is that in bacteria, the origin is sequence specific. So there's an actual sequence that says, haha, start here, right? Eukaryotes don't have that. So we are not sequence specific. What that means is our origins can float around. So there's not a specific place where you start. You just kind of start. We have a lot more going on in our initiation of replication. There's more factors that are associated with the helicase and the primase complexes. So that's always usually the case with eukaryotes. We oftentimes add a lot of extra stuff to it. A good example of that sort of extra wrinkle in the primase is that instead of just having one primase like bacteria have with the RNA polymerase, what we have is an RNA polymerase, but we also have a DNA polymerase as well, which is unusual, but we do. And so that kind of helps to improve our priming rates. Think about it. If you've got multiple replication bubbles scattered all over your genome, it's probably a good idea to have a healthy stock of primases because you're going to have a lot of priming sites to set up. Also, the polymerase itself has a few differences, right? We talked about the beta subunits, the clamps associated with bacterial replication, and DNA polymerase has some specific subunits that are usually fairly of a fair amount of importance, right? We have subunit epsilon and delta. So these are both subunits in eukaryotic polymerase that have major functions and major roles in the overall complex. Now, I don't want to get into the weeds on talking about the minute functions of epsilon and delta and alpha and beta and gamma and all that sort of stuff, right? So that's a tale for a different class. Like if it was a molecular genetics class with a very detailed and weedy replication segment, like a higher level eukaryotic class, we would get into that. But that's not our case here. So let's talk about the ends, right? So the ends are a little tricky. And at the end, you're going to find structures called telomeres. And so basically what telomeres are like adapters of repeating sequence. And what they do is they protect the ends of chromosomes. They protect it, first of all, from nuclease degradation, because remember, whenever you have an open end, that's like an exonuclease saying, aha, there's something for me to chew onto. So you protect that with your telomeres. Because the last thing you want is for your nuclease to go in there and start chomping up a gene that you need, right? So if you just feed it a bunch of repetitive DNA units, who cares about that? You can chew up that as much as you want, right? That's kind of like having the dog chew up their dog toy, as opposed to the couch or the wall in my dog's case, who actually chewed a hole in the wall, like an actual wall. I was like, I can't even get a hole with a hammer in that wall, and she managed to get it with her little muzzle. She's in Australia, part Australian Shepherd, so that's why they're little. Never trust the blue eyes, right? They sucker you in, but that means they're nuts. But she's cute and cuddly, but she's crazy. So they also protect it against shortening. Not the stuff you use in baking cookies. Sorry, I'm a little hungry. That's why the cookies came out. But anyway, but this is actually chromosomal shortening, right? Because if you think about it, every single time you go through a round of replication, the lagging strand causes a problem, right? Because when you're looking behind you where the new template is, at some point, you're going to run out of template. That means you're going to come to the end. So what that means is you lay down that last primer at that very end. And then when you remove that primer, guess what? You have no way to fill that in, right? Because you need a 3' OH and it's not there. Why? Because there's nothing beyond the end. And so it shortens your chromosomes. And of course, that's a bad thing because after every round of mitosis, if you're shortening your chromosomes by a little bit, eventually, by the time you've done as many mitotic rounds as us, you'll have no DNA left. This is what it looks like. So leading strand, not a problem. Lagging strand, it is, right? So here's your last primer. Of course, your Pol 1 comes in and does what it needs to do, basically removes the primer. But the next thing it needs to do is lay down a replacement for that, right? But can it? No, because out here, there's no 3' OH. You're at the end of your line. So there's nowhere for the pol 1 to attach to in order to fill this in. So what happens to all single-stranded nucleic acid? The destiny of all single-stranded nucleic acid is degradation. If you love the game Pac-Man, then you'll love replication, okay, and nuclease biology. So what happens? You basically just chew back this overhang, and you've got a shortened piece of DNA. Eventually, if you continue to shorten, that's going to run into some necessary essential genes, and basically you'll be a genetic train wreck, probably before you get out of the first trimester. So nobody would be born, basically. There would be no human beings on the face of the planet if we didn't have telomeres. So let's take a look at them, right? So telomeres, up close and personal. So these are basically repeated sequences of DNA that can be reproduced by an enzyme called telomerase. So telomerase basically will take these repeated units of DNA and just add them to the end of the chromosomes, thereby lengthening the that end. And so a telomerase, unlike polymerase, is a self-starter. That means it has everything it needs to basically lay down these repeats. As a matter of fact, one of the important things to understand is that it actually has its own template. It's an internal RNA template that it uses to lay stuff down. So it doesn't need the DNA template. All it does is basically use its own internal template, and it just basically copies that template as many times as it wants. And it basically just extends itself off. So what this would do then is this basically has the effect of lengthening the telomere. So telomerase will lengthen your telomere, thereby protecting you against chromosomal shortening. Now, as it turns out, what we've also noticed is that telomerase is not only heavily regulated, but it's also related to senescence. It's actually directly proportional. What does that mean? It means the longer your telomeres, the longer the shelf life of your cell. So that's what senescence is, right? Basically the shelf life of your cell. As a matter of fact, what we noticed was that if you create telomeres on a cell that are long, like artificially long, then you can prolong the number of rounds of mitosis that cell can go through, essentially, effectively, extending its life. Okay. Now, there's a lot more to cell senescence than just telomere length. This is mostly attributed to mitosis, right? Cell reproduction. And so this obviously then is a big deal, right? Because what this is, so you don't want telomeres that are so long that your cells never die, right? Because your cells will still break down and go through wear and tear. So the last thing you want is an immortal corpse, right? Which is the reason why the quest for immortality is a fool's dream. Because I don't know about you, I mean, by the way, immortality is oftentimes pursued by the young and the stupid old. Why do I say stupid old? Because when old people are on the quest for like the fountain of youth and immortality, they don't want to be immortal in the body they've got. They want to go back to when they were 20 and they want to be immortal that way. Well, let me get a news flash. If you're like getting older and you become immortal, guess what? You're just not going to die. You're going to become like a walking corpse. Ask anybody who's aging. And one of the worst things you can do is basically continue aging forever. So there's a reason why old people come to a sense of peace about death, because a lot of times it's like, you know what, that's actually a better deal at this point. So I can't imagine anything worse than being alive on this planet indefinitely, with as sucky as it is. So that's basically why you have this heavily regulated, right? So it's like, you don't want your cells living forever. You want them to last long enough to be functional, but you want to be able to tightly regulate how you build out these telomeres. So as a result, telomerase is heavily guarded and heavily regulated. Well, here's the thing. When you take a look at telomere length and senescence, that basically gives you essentially an immortal cell. Who do you think is going to be interested in that? Yeah, cancer cells, right? Because remember, we already talked about in chapter 10 how cancer cells already take the adult out of the room, right? P53. That allows them to be able to divide regardless of whether they have a train wreck for DNA or not, right? But here's the problem. If you're like a genetic train wreck, then you're going to become non-functional and you're going to die eventually, right? Even like with normal telomeres, you only have like a certain period of time. You only have like a certain shelf life, and it's set by your telomere. So you can only divide so many times before you die, okay? That's entirely unacceptable for a cancer cell, right? Because what they want to do is they want to be able to divide however they wish, right? Get rid of P53 and as often as they wish, for as long as they wish, right? So oftentimes what will happen is you'll see in cancer cells, not just missing P53 because you're removing the adult out of the room, but you also see the activation of telomerase, which basically creates immortality. Which is one of the really big landmark innovations that cancer cells try to make in tumor progression. Right. So the first one is getting rid of the regulators, getting rid of the adult supervision. That's the first big step. The next step is finding your path to immortality. Basically divide as much as you wish and you'll live for as long as you wish. So you can accumulate mutations and never die. And the third one is create your own blood supply. It's angiogenesis, right? Because the other limiting factor to your tumor growth is outgrowing your supply lines. But if you figure out a series of DNA mutations that allows you to be able to grow your own blood vessels, then it's like you can create your own supply line and you can grow as big as you want. This is the reason why some of those teratomas get really big and you see things like teeth in them. That's what's happened. Basically, you flipped so many switches genetically that you've basically reignited the developmental pathways for teeth production or for hair production or crazy stuff. I mean, you see all sorts of crazy stuff in teratomas. Yeah, that's why you want to get, I mean, it'll turn into another mini me, only like a train wreck of a mini me. Right? Yeah. So here's telomerase. So here's your short piece. Now, typically speaking, you'll chew back, right? You have kind of like that little bit of a piece of an overhang there. That's usually from the telomere. And so what telomerase has then is their internal template, which is this guy right here. So it'll grab onto and anneal to that last little chunk, but then it'll just start filling in from its template. And then what it'll do is it'll shift down and it'll basically re-anneal at the end of your telomere, and it'll just basically fill it in for as many times as you want. So you're basically just creating one repeat after another, to a certain length. And then of course it becomes easy then at this point to just basically fill all this stuff in. So you just kind of fill that in and then you're good. Right. So that's basically how telomerase works. Now, what happens if you screw it all up? Like really, really badly? Well, it gets us to DNA repair, right? So we already talked a little bit about repair. Like if you just flub it up, like if you just do like a little bit of a typo, then of course we have proofreading. So this will be like an error of replication, kind of like when you're typing and you mess up the word "the," which I can seem to never spell right the first time. So I'm always backspacing that one and retyping it, right? That's what the proofreading is. It's just basically to correct those really small errors in replication. But what happens if there's an error or a damage in DNA that doesn't have anything to do with an error or slip up in replication? It's because of an outside insult, right? So we have a lot of those actually. That's probably where the majority of our mutations come from, is from outside insult, because our mechanisms to sort of proofread and to fix our errors are actually pretty good, and they're pretty tight. So if it's an outside insult, you're talking about what's called a mutagen, right? So basically a mutagen is anything that will increase your mutation rate above background level. So you have a normal mutation rate that's always constantly going on. So you're accumulating mutations right now. For most of you, since you guys are fairly on the youngish side, you guys don't have as many accumulated mutations as I do, because I've just been walking around on the planet a little while longer, accumulating more background mutagenic environmental stuff, basically. So what is the background? Well, it's simple stuff you don't think about, right? Just natural cosmic radiation, UV radiation, just stuff you're always exposed to, just walking outside, right? So there's radiation associated with that. In Colorado, part of our background is radon, right? So we have a huge radon issue. It's a soil isotope that decays into a gas. And for the most part, just like walking around, just out on trails and things like that, it's not a problem because it's a gas, so it dissipates in the atmosphere readily. But that is a type of background radiation. The reason why radon's a problem in Colorado is not because of the fact that it's there in Colorado. It's in most soils in Colorado. Is because a lot of us may have houses or plan on having houses that have that really nice finished basement, the man cave, or whoever's cave it is, right? Or the bedroom. A lot of living space houses in Denver, I've noticed, right? They finish the basement, put a room down in there, a full operational bathroom, and you call it a 4-bedroom instead of a 3-bedroom. Well, here's the problem with that. If you do that, if you look for a house like that, then look closely, find the crawl space. If the crawl space is down there in the basement and it's open, then you'd better insist on radon mitigation from the seller. Why? Because the open crawl space in the basement is soil, it's open soil, and there's radon naturally decaying from that soil. The gas of the radon, the radioactive gas, is accumulating in the basement to abnormally high concentrations above background level. So somebody who's living in the basement will be basically breathing in abnormally high amounts of radon, and their frequency of lung cancer will probably be considerably higher. Okay. Which is a reason why in any house in Denver, check your crawl space. And if it's not mitigated for radon, I would suggest you test it. And if it's high, then I would suggest you get it mitigated if you plan on spending any time whatsoever in the basement. It's pretty common. By the way, that's something sellers should do. If you're a buyer, insist on it because it is a problem in Colorado. That's what we did. So if you've ever seen like houses where they got those really hideous looking little white pipes on the side of the house sticking up like a little chimney, that's a radon mitigator. So that's basically, it's very simple. All they do is just put like a white tarp over the soil crawl space with some ventilation hoses and things like that. And it just basically ventilates the gas up through that little white pipe out into the atmosphere. It's very simple.
Oh, so it's just not sucking in.
Yeah, so it's not, so it's not accumulating in your basement. So, and they've got, a fan basically going like all the time. It never stops. It's like constantly sucking that radon out of there. So that's an example of just a natural, in this case, a natural source that would have the capacity to rise above background levels. Usually speaking, that's an example of radiation, right? So most of these mutagens that we run into are the radiation or their chemicals, right? So and for radiation, it's a little difficult, right? So in Colorado, the radon is the best example of something that we would just dumbly fumble into and expose ourselves to. But for the most part, in other areas, other states that don't have radon as an issue in their soils, to get radiation poisoning, you'd have to be in the wrong place at the wrong time, right? A little too close to the cesium chloride source in a research building, or making the stupid mistake of taking just a nice Sunday afternoon stroll through Chernobyl, right? So that would be a bad idea, or some other kind of a nuclear incident, right? That's the issue with radiation. So something bad would have had to happen for you to get that kind of radiation exposure usually. Chemicals essentially are like all these mutagens and carcinogens, right? So we always see the California warnings has been known to cause cancer in laboratory mice in California. Well, they're talking about a chemical that's in that particular food stuff that's been known to cause problems in mice. And so obviously, if you're ingesting large quantities of that, then you're getting a relatively large dose of that chemical, which could cause problems. I mean, if it's something that you maybe ingest once every blue moon, then probably you don't need to hit the ceiling just yet, right? So there is kind of a dosing situation. But these are important sources. Now, what kinds of repair systems do we have? We have lots of them, first of all. We actually have an entire chapter in the genetics class where we talk about repair and mutation and repair. There's quite a few of them. But for our purposes, we can categorize them in two major categories. You have either specific repair mechanisms or non-specific. And in specific, it basically is kind of what it says it is. So you're looking for a very specific type of damage. And you only repair that one, right? So if there's some other type of damage that's not your thing, you don't even touch it. Now there's non-specific one where you're basically looking for multiple. So this is like a general, looks for general damage. Just something that looks wrong. Don't even know what it is. Don't really care really. It's like, this is just wrong. It doesn't look like it's right. You just basically go after it and you fix it. So we'll take a look at a specific type of repair, and then we'll take a look at a non-specific type of repair. The specific one is what's called photo repair. So in photo repair, what you basically have is DNA that's out there getting blasted by UV radiation because you are at the baseball park in right field during a day game, being miserable because the Rockies have only won like 7 games this season. Right, almost as bad as the A's. I've only got 5, both my teams. So I don't even have a decent team ever. It's like my fanship is like the kiss of death. So just for you Avs fans, I resist being an Avs fan for your sake, so that the Avs can actually continue winning. See, by that metric, I should change from being a Raiders fan to being a Broncos fan, because then I'd like, poison the Broncos. That'd be very Raider-like, wouldn't it? That'd be very Raider fan-like. Yeah, that would be. I can't do that. I can't do that. I just can't wear blue and orange. I just can't do it. It's like why I don't own a lot of red in my wardrobe, because I don't really mind the Broncos, but the Chiefs, oh my gosh, it's just like a ketchup and mustard hot dog explosion all over the place. I just want to strangle everything that is red and yellow. So I wear nothing that's red. Actually, all the teams I hate the most are red. The Niners, I can't stand the Niners. Even worse than the Chiefs, actually. Yeah, the Diamondbacks. Yeah, no, can't do that. That just makes me want to vomit. The only red team that I have a soft spot for is the Cardinals. That's it. Everybody else is right there in the pit of hell for me. Okay, that's it. Now that you know my allegiances and how they break down, you know who I'm going to be voting for. Okay. So what happens when your DNA is absorbing all this UV radiation? Well, what happens is typically in your DNA, you'll run across a situation where you have 2 thymines in a row, right? And you know that those thymines are associated with adenines, right? But what happens is when UV radiation hits these guys, it causes the thymines to actually cross-link with a covalent cross-link, causing this sort of thymine dimer. That's basically what this guy is, a thymine dimer. Well, here's the problem. When you go to replicate that, your replication machinery doesn't know what to do with this covalently linked thymines. So it's looking at this thinking, what is this? Right? So I got to guess. And so when it guesses, it oftentimes guesses wrong, which then causes a mutation, which then would cause cancer. So as it turns out, in bacteria, you have an enzyme called photolyase, which has the capacity to actually fix this problem. So it's basically activated by blue light, which is pretty cool. And so what photolyase will do is basically it will activate in blue light, and then it'll actually cleave the covalent bond that's connecting the thymine dimer. And then once you cleave that bond, you're good. You basically just smooth it all out. But there's a problem. That's in bacteria. So when you take a look at the mechanism, it looks like this. So here's your 2 thymines next to each other. When UV light hits it, it's going to create these 2 little covalent bonds in there. That's your thymine dimer. Then photolyase, which is blue light activated, will come in there, notice and recognize the damage first of all, and then it'll basically cut that covalent bond and smooth it all out. The problem is this: photolyase is not in humans. So what do we do when we're out there sunbathing, accumulating thymine dimers and adding to our DNA damage repertoire?
How we were the smart ones.
Well, not in this case. In this case, bacteria have a speed, right? How do we solve this problem? We don't. Oh yeah, sunscreen initially, right? But once you have the thymine dimers, there's no way to fix it. So we basically live with thymine dimers pretty much indefinitely. We have no way to undo a thymine dimer. We don't have photolyase. We don't have any kind of an enzyme that's even remotely close to it. So we have nothing that can actually help us resolve the actual dimer itself. As a matter of fact, when we cover this section in the genetics chapter, we kind of go over some of the little tricks that humans use to work around it. That's kind of what we do, is we do all sorts of crazy little recombinational things, like little weird kind of patchwork sorts of things to try to work our way around it. You know, recruiting sisters from another homolog to try to help rescue us, that kind of thing. So there's lots of little weird things that we do recombinationally, but ultimately that dimer is still there on that strand. And so obviously sometimes that process is error prone because you're trying to do your best to sort of salvage the situation. The more of these you have, by the way, the more likely it is that you're going to create mutation. And you don't get rid of them, right? So they're always there. So even if you got it right this round, guess what? The next round, you got another shot at it to get it wrong. So it doesn't, like, you don't really ever fix it. It's always going to be a problem every round of mitosis for this particular section, Cadence.
Would that be like black lights and UV black lights? Are those like dangerous?
Yeah, as a matter of fact, for most black lights, depending on their strength, they have different strength black lights. Some of them are actually just more like purple lights or indigo or violet lights, but they're not true black lights, so they kind of diminish their UV emission. But a black light, like the older black lights, like from the 60s, they were true UV emitters, and there would always be a label on there that would say something like "never stare directly at the light." Why? It's not because the purple is going to harm you, and it's not really anything that you can see that's harming you. It's the UV that you cannot see that's basically entering into your, especially with your eyes, because your eyes have no protection, right? The UV radiation will go straight through your pupils, which is basically an open hole, directly to your retina and can damage your retina, right? So that's in like an almost an immediate assault, an insult on your retina. That's the reason why they say don't stare at the black light.
So if I'm shining like a UV light on something that is UV reactive, and I'm staring at the UV reactive thing, is that like?
No, that's just normal light. Because usually when you have the black light, which is kind of a purplish color, right? Then what's usually going back at you is further up in the rainbow. So it's usually, you know, red, orange-ish kind of color, which are lower energy lights. So once you're looking at those, all you're doing is looking at orange and red light. So if I shined it on, you know, orange fluorescent paint, I saw the orange glow, it wouldn't be any different than if I were looking at Antonio's orange shirt.
But if it's something that has like uranium in it, is that?
Well, that's different because you don't need a black light for uranium to come after you.
Yeah, it's uranium.
That's-
gas is what
So that's a different thing because uranium is emitting alpha particles, which is a type of radioactive decay, along with radiation. So that has nothing to do with black light because any kind of a radioactive isotope is an emitter already of radiation, and that's always going to come after you. And that's not one where it's just like, don't look at it. That's one where it's like, don't be around it.
But what if it's just
So-
really low quantity and it's very old, like over 100 years old? Is that like still? Should I just stop keeping this stuff in the house?
This is very oddly specific.
Yeah,
I would,
uranium.
yeah, it's
I
like,
want uranium
so wait a minute,
gas, I'm just trying to
I'll
suss out
write
like-
some notes down here. So are you purifying the uranium to weapons
No,
grade?
it's
No, I was kidding.
uranium blast. It's just blast from the 1800s and they put uranium in as a coloring.
Yeah, kind of like when we used to make the glow in the dark watches
It's
with
not- it's
radium.
not- it's
Yeah.
like way less dangerous than that.
Yeah. In that case, if you're talking about quantity and it's just a little bit of it, you're probably okay.
Yeah, it's UV reactive. Like you shine the UV.
I mean, if
light
you're
on it,
concerned, I mean, you could like, if you have a large collection of it or something like that, like for instance, in geology, where you're working with natural isotopes of or isotopes of potentially radioactive minerals and things like that. Any kind of a geology lab would have a Geiger counter along with it, just so that you can monitor how many hits you're getting per minute. And you know, there's a range there for safe. And it's always a matter of not just safety, but like exposure, like how much time on exposure. And if it's really a problem, then, you know, they kind of develop this whole badging system where they can actually monitor and quantify just how many REMs you're actually getting access to. But in the case of something like that, which is along the lines of a glow in the dark watch from the 60s, then you're probably not talking about a significant source. I mean, if it's something you were worried about, I mean, you could just put it like in a lead containing container, just for, you know, my guess is it's probably not worse than radon, actually. Good.
Do reptiles have the same problem? Do they have to work around it since they're cold-blooded, or do they,
Yeah, so I mean, everything was going to have repair in them. So it's not really linked to cold-bloodedness, because cold-bloodedness is just about whether or not you have a core body temperature and what that is. And that's just a different way of doing things. And so you're going to have DNA repair one way or the other. So this is actually more fundamental than bloodedness. So whether you're an exotherm versus an endotherm.
well, I was just saying because they're on the sun so much to warm their body. I didn't know. If they,
Well, I mean, usually when you have something like that, I mean, there's a couple of things. Number one is your life cycle isn't long enough to really matter, because by the time you'd accumulate enough mutations, you've long since been dead. But reptiles can last quite some time. They can be quite long-lived. And in which case, they would probably do the same thing as us. They'd probably accumulate pigments that would have the ability to kind of combat or shield off UV radiation. And they probably also have repair systems just like us. So, because it's a eukaryotic thing. It's not like a mammal thing. So it goes even further back than just that. So
it's like all of you.
yeah, it's very, very fundamental at this particular point. So there's for the non-specific repair, what we have is essentially just general damage that you're recognizing, and you have typically a three-step system that's pretty true for all DNA repair systems. So you have damage. You have to recognize the damage first of all, right? You have to sort of recognize it as, okay, this is all screwed up. We need to fix this kind of a thing. And then, of course, the second piece is you have to have the capacity to actually remove it, right? That's the excision part. So you have to be able to excise it or cut it out. And once you cut the bad part out, then you have to be able to sort of lay down the new, right? The idea being that whatever damage you had was probably due to some random insult from outside. The chances are that random insult was periodic, right? And it's probably not a continuous or chronic exposure. So therefore, the chances that you're going to make the same or get the same amount of damage in the same place is remote. So you're going to lay it down again, and this time it'll be correct. Yeah, that's kind of what you're looking at there. That's kind of how it works. So here you can see some sort of a just general damage sort of a situation here. So you can see it always creates some sort of an anomaly in the structure of the DNA. And then your DNA repair enzyme or complex in this case, will actually come in, recognize the area of damage. In this case, we're looking at the UVR ABC complex. A, B, and C are three different domains that recognize, and then one cuts and the other one kind of lays it down. But UVR stands for UV radiation. And this is the enzyme that will basically recognize it. It cuts it out. And then basically you just lay back down the new segment. You fill in the gap with a polymerase, and then you're fine. Okay. Now, there's a couple of different outcomes from this, right? So you have, in terms of non-specific, you have kind of two main global scenarios. The first one is what we refer to as error-free, like DNA breakage is an example of this, right? So for instance, if you have a double strand break, this is easy because all you have to do is just basically seal the ends together. You do that, no harm, no foul. There's no error there. It's a super simple, easy one, easy fix, right? Low error rate on that one. But if you have a lot of damage, a lot of massive damage, basically it's less likely to fix it all. So the more of it you have, then the more problems you have to deal with, the more you're having to sort of put this together, which means the more likely that there's going to be damage that's basically not fixed, right? So you're more likely to lead to errors in this particular case. Okay. So that is the end of 14. Now on to 15. Okay, so in 15, this is our last chapter, by the way, of the semester. So this is basically taking a look at how genes work. Now, when we take a look at how genes work, it's basically all captured under the umbrella idea of gene expression. And generally speaking, when a gene expresses itself, it expresses itself in the form of the protein that it encodes, right? That's basically how it expresses itself. And so there's a lot that goes into that. But as always, we're going to start every chapter along with our history lesson, right? So you get a good healthy dose of history as opposed to just hardcore science, right? Get a little bit of everything in there. So let's go all the way back, right? So we kind of, with replication, we kind of started at the beginning, kind of moved forward to about halfway through the century. This one, we're going to go all the way back again, back to the 1900s in 1902. Now, remember, at 1902, this is the point at which Mendel was just being rediscovered. So William Bateson, William Punnett, those guys were just rediscovering Mendel's work. They were starting to do Mendel's experiments in their own systems, whether in 4 o'clock plants or if they're in corn or if they're fruit flies, right? Some people were studying humans. And we talked about that a little bit, right? In the last chapter, when we were talking about pneumonia and things like that, right? With Frederick Griffith's work on the mice and that sort of thing. Well, Archibald Girard was actually before Griffith, because Griffith was 28. And so Girard was basically a good 2 decades before that. Not only that, but Girard was like right in the middle of that Mendelian rediscovery. So the important thing about Girard's era is to remember that he wasn't that far ahead of Mendel. Remember, we were just rediscovering Mendel. We were just wrapping our heads around Mendel's data, seeing what it looked like in other situations. But you still really didn't know what a gene was. You still really didn't know what chromosomes were. Mitosis and meiosis was fairly innovative, and there's still a lot of questions associated with that. So a lot of the things we take for granted in chapters 10, 11, and 12, Girard still didn't know. So he wasn't really that far ahead of Mendel. So what Girard was doing was he was studying a specific human disorder, a metabolic human disorder called alkaptonuria, which is really cool because the metabolic disorders are really simple. Right? Basically, you have reactants, you have products, and you have an enzyme that drives it. Right. So what he noticed, however, is a couple of things. First of all, he knew that his disease was caused by the lack of the factor. He probably didn't really understand enzymes the way you guys do. But whatever this factor was that drove this chemical reaction, this sort of enzyme that drove this chemical reaction, he knew that his alkaptonuric patients did not have this, right? So that part he knew. The other thing he also knew from Mendel's work because remember, he's reading through Mendel's stuff. So he's reading through the monohybrid crosses. He's reading through the dihybrid crosses. And as he's looking, he might have even been sort of consulting with, folks like Bateson and Punnett and talking to them and say like, well, what are you seeing in corn? It's like, what's your big pattern? I mean, how many have you done? What's your overall pattern? And so he knew what the pattern of recessive inheritance looked like. So when he looked at Mendel's work, he could see those ratios. He could see the alleles, the predictions. He could see all this stuff coming together. He saw the pattern of what it looks like for a recessive allele to segregate through multiple generations. And you know what else he saw? He saw the same pattern in his families. Now, the important thing about this is, first of all, he knew 2 things. Enzymes drove his reaction. That was what was at the problem, the core of alkaptonuria. The second major thing that he noticed was that this disorder is segregating in the same way that Mendel's traits were segregating. He saw the same pattern of recessive inheritance that Mendel saw. So he made the connection between genes and enzymes. Now, he didn't know anything about how you code for enzymes and things like that, but he made this connection that his enzymes, or lack thereof, if you will, was showing a pattern of inheritance that was something that Mendel saw. Now, did he go out there like Mendel and take purple flowered people and white flowered people and cross them together? Did he create true breeding strains of people? No, right? You can't do that. But guys like Bateson and Punnett were, right? They were creating true breeders of corn, true breeders of 4 o'clock plants, true breeders of Drosophila, true breeders of mice, or whatever it is that you're looking at, right? And they were getting all this data and they were seeing all these patterns. And then Girard basically applied that to his system. And he said, you know what? I can't produce true breeders, but here's what I can do. I've got a patient with alkaptonuria in them, and it's segregating to their family. So what I can do is I can get to know the family. I can create a genealogy, right? So we've known about genealogies forever. We were really good at that. Not poor people, but rich people, right? Because that's how they figured out who gets all the money, right? But basically, you knew how to do genealogy. Well, in this case, instead of doing a genealogy to prove your ancestry, you were doing genealogy and overlaying the phenotype, in this case, alkaptonuria, on top of it. Because the question was, well, wait a minute. If we can use genealogy to prove ancestry, then shouldn't the phenotype of alkaptonuria also be predictive from the genealogy? And that's exactly what he saw. So what he created was a pedigree. He basically sat down and said, okay, you have alkaptonuria. What about your siblings? Do they? How about your parents? Do they? How about your uncles? Do they? Your cousins? Do they? Right? All the way back, as far as we can go. And then what he saw was this pattern emerging in his data. So he didn't start here and move forward with crosses, which is like what most genetics do. He basically started here and he went backwards with already existing families. Okay. So what he actually did, without even knowing it probably, was what was the beginning of human genetics. Kind of the way that we do human genetics. Okay. So that was a big deal because that was early. So to link genes when we don't even know what a gene is, right? Remember, we started that conversation up a couple chapters ago, didn't we? What is a gene? We don't know, right? We don't even know what the genetic molecule is at this point. But he was linking this idea of genes to enzymes. Okay, obviously, Girard was part of that. DNA is the inheritance molecule group, right? Okay, let's fast forward a few decades to Beadle and Tatum. And of course, you see a date there, so you know what I'm going to do. Never miss on this one. Yeah, what's significant about 1941? At least for the US anyway. December 7th, 1941, a day that will live in infamy. Yeah, Pearl Harbor, right? That's a quote out of, that's actually the way FDR started his response to Pearl Harbor, right? This is significant, right? Because this is what dragged us into the war. Before then, we were basically just sort of on the sideline, just sort of putzing around, like somehow it didn't matter to us. And this eventually is what pulled us in there, right? So this is what got in here. This, in a lot of ways, a lot of people also look at this, like economic historians look at this as like this is the thing that pulled us out of the Depression as well. Yeah, FDR had a lot of interesting programs, and that kind of helped some things, but there was still very much an active economic depression going on at this point, and the industry of war really up-ramped production and changed the economy in a big way, and that's kind of what created. They probably took a lot of things actually in World War II. Yeah, bad thing. Well, they did cocaine too. I mean, they used to mix that with Coca-Cola. Yeah, so they probably did a lot of things back in the day. A lot of those, yeah, you used to get those like in just the local pharmacy, you know, just, I mean, stuff that was, I mean, because cocaine was actually just an over-the-counter kind of a thing back in the day. It was like, and then it became prescribed. Yeah, and now it's like illegal, so it's, yeah, it's kind of, yeah, nice.
This was even illegal back then. That's surprising.
So that's what was going on in the US, right? So here we go into the war. By the way, at this particular time, Europe was pretty well devastated. Most of Europe was learning German. Or you could say Hitler was learning French so he could communicate. Actually, he didn't care. He was just killing them all. But pretty much most of Europe was under Nazi control at this point. Most of the South Pacific was under Japanese control. What were the Chinese doing? Well, this is right around their Boxer Rebellion era. So they had issues of their own. And they were certainly no match for the Japanese at this particular point. Which is the reason why the relationship between China and Japan is very tense at best. That's actually an understatement. And actually China and the other Southeast Asian countries as well. Korea, for instance, that was a huge flashpoint as well. But that's all coming out of this. So here comes Beadle and Tatum. And so what they wanted to do essentially is they wanted to, instead of just following natural variation, which is what Mendel did, right? So Mendel just said, okay, here's pea plants. I see purple flowers, I see white flowers, and I'm just going to study their genetics, right? So I'm just going to take what they give me and I'm going to study their genetics. And he came up with all these patterns of inheritance. And that's kind of what most of the people in the early part of the century were doing as well. But here's something in Beadle and Tatum did. What they wanted to do is they wanted to create mutations. And the question they had is that do the mutations themselves follow the same rules as the normal gene, right? So for instance, if I, instead of following purple, like what if I can't get a hold of purple, you know, can I mutate this gene, like, and my mutant would cause it to be fluorescent pink? And then would that fluorescent pink mutant phenotype follow the same rules that the normal gene would? We didn't know that until Beadle and Tatum showed that it does. Why is that important? Because what that does, it opens up our ability to study these systems. Why? Because natural variation will only get you so far. What we're really good at is screwing things up. That's what we're really good at, right? And we're really good at blowing things up and learning how they work by how they screw up, right? So like if you're on a team and you have a particular function and I'm trying to figure out what your function is, it's harder for me to study the whole team as a normal functioning unit. The better thing to do is like, if I just take that one person out of the mix, and then that's like my mutant. So it's like an abnormal scenario. Then I take a look at see what's going wrong. Then I can see like, oh, okay, so then you must have been doing this and this and this because you're not able to do that anymore, right? So or this is how it went tragically wrong. So now I can understand what you were doing and why your role was so important, right? So we're good at that. Mutations give us that ability. So we can take something, mutate it, which basically abolishes its function, and then we can study what its function was by understanding what's missing in that particular scenario. And so this is something that we always do. And so what they did is they wanted to create these mutations and follow them, and they did in Neurospora crassa, which is basically a fancy party name for bread mold. Right, that's what you do to impress your friends at parties, or not, right? That's the thing. But anyway, what they did is they used X-rays to damage the DNA of Neurospora crassa. So what this basically does, it induces mutations. Just blast them, essentially. And so you're going to be raising radiation levels above natural background, which is going to increase your mutation rate, which is going to basically mutate everything. And this basically causes mutations in random genes. So you're going to be mutating not just your gene of interest, but you're going to be mutating everything, including stuff you don't care about, right? Then what they looked for was some change or some sort of a phenotype. In this case, there was a selection process. And you're only interested in your phenotype. Here's a good example. At UC Davis, one of the things I did as an undergrad was I worked on a mutation screen for, it was a meiosis lab, right? So we were looking for mutations or genes that were involved in normal meiosis. And what was our marker? to show us a meiosis defect? It was non-disjunction, right? So remember, non-disjunction is that sort of unbalanced meiosis. So our big question was, what causes the balance, right? That whole balancing thing of meiosis, right? If there's a bunch of proteins that are doing that, then there's genes behind those proteins. And if we mutate those genes, then we can selectively screw that up. And then you're going to get more amounts of non-disjunction. So what we did was we just blasted these flies with mutagen, and then we screened them for excessive rates of non-disjunction. And then when we found one, then we'd select that female, and we would sort of preserve her into a mutant strain. We say like, oh, here is a defect in non-disjunction, our first allele, right? So whatever that is, we don't even know what gene it is. We just know that whatever is screwed up in there is affecting meiosis, and we want to know what that is, right? So then we'd say we had a whole catalog of mutations like that. But you had a selection process, right? You mutagenized it, which did global damage across the genome, but you only selected for one phenotype. You didn't care about the rest, okay? Now, what they did was they looked for nutritional mutations. Now, the important thing about Neurospora is Neurospora can make its own amino acids. Which is cool. We can't, right? We have to ingest our amino acids, but they can make their own. So because they can make their own nutrition, they don't really need a lot to grow on, right? So if you were to put a normal Neurospora on minimal media, this basically supports growth for normal, healthy individuals because they got all they need, right? However, if you were to mutagenize one and one of these mutants was in a gene involved in making amino acids, then all of a sudden you wouldn't be able to make everything, right? So won't support mutants in essential amino acid genes. And of course, you're interested in those genes. Now, what they were looking for was a specific one, however. So they wanted to look for any gene that was in the biochemical pathway in the production of arginine. They could care less about lysine, they could care less about glutamate. They just wanted to see what genes are involved in the arginine production. So those are the ones they worked with. And they were able to basically identify the pathway of enzymes in this particular production scenario. So what they kind of did is they modified Gerard a little bit. It wasn't really a hard and fast linkage, but he was kind of like the first tease a little bit. You notice scientific discovery always starts out the same way, doesn't it? There's some sort of an experiment that's not quite compelling, but it's just tantalizing. And then eventually, somewhere down the road, somebody has to do a little bit more of a lockdown experiment or a series of experiments before finally it's like, okay, now we're good, right? So it's the same basic pattern. So what they actually figured out was the one gene, one enzyme linkage. So that is to say that genes indeed are responsible for making enzymes. So let's take a look at their experiment. So in this particular case, what we're going to see is they're going to take wild type Neurospora, which is just normal bread mold. They're going to blast it with their X-rays. Now, remember, in the X-rays, you're doing global damage. So it's mutating all sorts of genes, not just yours. And then what you want to do is you want to grow it on rich medium. So this is basically luxury. So one of the things you always do in a mutagenesis screen, like whenever you mutagenize something, either with radiation, which by the way is what we use cesium chloride for. I mentioned that before as a radiation source. You'd only see cesium chloride in like a research university as a source of radiation to mutagenize things like this, to generate mutations. But basically what happens when you mutagenize them, so when you get blasted with X-rays, it basically just, it just really beats up the cells. It really beats them up. And they're just kind of like limping along, like, they're just like they're just bloodied and bruised. They're like literally like right on the edge of life. So what you want to do is you want to beat them up, push them up right when they're kind of teetering on the brink. And then what you want to do because they've got a lot of mutations in them now, right? Then what you want to do is you want to bring them back. That's why you put them on rich media. So give them everything they want. Give them the entire Thanksgiving spread, right? 12-course meal, the whole deal, everything they want. They want a hot tub, yours, right? They want a mansion, yours, right? They want a yacht, yours, right? This is the lap of luxury. You have everything. The idea is come back. Don't die, because we want your genes, right? We want your mutated genes. Come back, recover, lick your wounds, heal yourself up, become strong again, but hang on to your mutated genes. That's what the growth medium, the rich medium, is for. You're restoring them back to health. Then what do? Then you have to screen them. This is your selection. So how do you select for what you want, right? Or what you want is arginine. Well, here's what you want. First of all, if you plate these cells on minimal media, what's going to happen? Yeah, if you got a mutation in an essential gene, they're going to die, right? Okay, great. Let's say that you have mutations in a lot of essential genes. Then pretty much everything's going to die. Arginine included, lysine, glutamate, whatever. Doesn't differentiate what you're looking at. It's just, can you live with it or without it? Right, that's all you're doing here. Then in order to determine that it's arginine, what you do is what's called a rescue. So you take your minimal media, but then you add arginine. And if you add arginine and you get growth, then what that means is arginine was what you were lacking. That means you're an arginine mutant. Or what you're mutated in is in the arginine production pathway. So you could identify mutations that were in the arginine pathway by doing this selection process. But here's the problem. If you've got lots of players in that arginine pathway, you don't know which one's which. At this point, you don't know how many genes are involved. You don't know how many mutations. You could have like 20 mutations. It could be 20 different genes, or it could be one gene with 20 different mutations in it, right? So you don't know. So part of when you're characterizing a mutation is you're trying to figure out, well, how many genes am I talking about here? Am I talking about different mutations in a gene? And so there's a lot of characterization that goes into mutation. That's kind of what I do as an undergrad. Take a mutation that we haven't characterized yet and try to characterize it and try to figure it out, which is a lot of fun. But here's what they did. In order to screen this and try to characterize this, what they did is a couple things they knew, first of all. Now, this kind of has a few of the answers here, so I want to sort of highlight this. So what they didn't really know was this piece down here. What they did know, however, was that there was various molecules involved in the production of arginine. Obviously, they know arginine, but they know that arginosuccinate is involved. They know that citrulline is involved, ornithine and glutamate are all involved. They don't know the order, right? So they don't know that glutamate comes first and ornithine comes next. They don't know that, right? They just know that these are involved. That's all they know. So part of what they're trying to do is order them, like which one's first, which one's second, which one's third. That's part of what they're trying to do. And here's how they did it. So they did a series of rescue experiments, just like they did before. So basically when they add arginine, then because they're arginine mutants, you would expect everything to grow, which is exactly what they saw. So everything grows, that's their baseline. So they got that one. Good. Okay, now that basically means you're arginine mutants. Then what they did is they took one of their chemicals and they said, okay, wait a minute, let's go ahead and add arginosuccinate. So what this basically means is arginosuccinate is added and you have everything you need in order to get to the end, then everything should be fine. However, if you don't have everything you need to get to the end, I'm talking about the enzyme itself, then you should see no growth. So here's what they see. They see growth, 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 and then no growth. So that one is a failure in this enzyme, right? Because you don't have the ability to convert this to arginine. Now, then what they did is they added citrulline. The idea here is if you've got these two are intact, then you should be able to make arginine. However, because we added citrulline, if this one is bad, then you're not going to be able to get past this level. So what they saw was growth, growth, and then they saw a breakdown here. So in this particular case, this one represented a breakdown in citrulline converting to arginosuccinate, which is the reason why you didn't make arginine, which is the reason why they died, right? Then, of course, they added ornithine. Now they're getting further down. And then they noticed that, well, only in one of them are you able to actually live. So you basically are able to make it all the way through. But the rest of them, essentially, you stop growth. So that basically means that this one here, the first one they see, is actually a disruption in the ornithine to citrulline segment. And then, of course, finally, the mutation in the first one, glutamate, that's this one. So that's the one where it doesn't matter what you add as much glutamate as you want, you're never going to get there, right? So that's going to be this first one. So what they were able to do was order the events. So they basically said that if we mutate the arginine H, that is enzyme H, and it basically eliminates or debilitates that enzyme, then we no longer have the ability to convert arginosuccinate into arginine, right? If we mutate arginine G and we kill the enzyme there, then we no longer can do that one. However, if we add arginosuccinate, we can, right? Because this one's still good. Now, if we mutate F, then that means we can't convert ornithine to citrulline. But guess what? If we add citrulline, it'll make it. If we add arginosuccinate, it'll make it. And then finally, last but not least, if we mutate E, we're going to take out enzyme E, then we're not able to make glutamate into ornithine. But if we add ornithine, we can make it. If we add citrulline, we can make it. If we add arginosuccinate, we can make it. So you can see you kind of have this scaffolding effect where by taking all the look at your data, you can basically order them by saying, which one's first, which one's second, which one's third. How do we know that? Because we mutated the genes, which then disabled the enzymes. So from this, that's where they came into their one gene, one enzyme idea. In a lot of ways, this taught us how to study biochemical pathways. Now, at this time, before then, what we used to think was, and even after this a little bit, we used to think that most processes are like one enzyme, one process, right? So it's simple. What we now know is that actually most processes are not one enzyme. Most of them are driven by big complexes of many enzymes, like replication, for instance. And not only that, but a lot of systems are not just one step. They are a multi-step pathway where you got multiple pieces to it, multiple stepwise progressions, all of which require each other. So we noticed that the biochemical pathway idea was actually more the rule than the exception. At this point, we thought it was more the exception. Was there a hand going up back there, you saying? So the mutagens in this case is X-rays. So they did that early in the beginning. And then they generated all the mutations, and now what they're doing is they're sorting through all the mutations to try to figure out how they're related to each other.
By adding all the mutations together, then do we get arginine?
Well, so these aren't mutations themselves, but like for instance, if you added mutations together, then basically everything would be mutated, everything would be dead. Right? You wouldn't even get past. I mean, there would nothing would work. Right. So what they're looking at is individual mutations, but kind of in a series. That makes sense? Okay, so this kind of leads us to an idea in molecular biology that basically was developed by Francis Crick of diabolical fame. But I mean, to his credit, he actually did do some decent contributions to this particular era of molecular biology. But basically, he's the one who came up with the idea of central dogma. And the central dogma basically says just this: information in the cell flows from DNA to RNA to protein. So that's how it flows. So if you're going from DNA to RNA, it's called transcription, which I abbreviate XPN. If you're going from RNA to protein, it's translation, which I abbreviate XLN. So if you ever see me write that, that's what I'm talking about. Okay. Now, that's generally speaking. Back in the day, we didn't really understand retroviruses very well, but now we do have retroviruses, like coronavirus is an example of a retrovirus. These are violations of central dogma. In this case, they go from RNA to DNA, which is in a process called reverse transcriptase. So they make a DNA copy of their RNA. So it's got a backwards process. But for the most part, it is DNA to RNA to protein. Right? That's kind of your central dogma. And so this is for you picture people who like pretty pictures. Right. So if you take your DNA template strand, you make an RNA copy of that one instead of DNA. right? If you're making a DNA copy of the template strand, it's called replication. If you're making an RNA strand, it's called transcription, right? And then you use this code in order to make your protein during the process of translation. And that's happening inside the ribosome. Okay. So basically, when you take a look at transcription and translation kind of side by side, so you generally will only have one template strand read off at a time. So you don't have both sides read off. It's just one at a time. And of course, in RNA, you are putting in uracil instead of thymine. That's one of the ways you can actually determine quickly if you're working with RNAs, if you see a bunch of uracils in there. So a bunch of uracils will tip you off, but that's it. And of course, then the mRNA, which would be a copy of your DNA, will be used to make polypeptides. So how to choose and select individual amino acids when you're going through the production of primary structure. Okay. Then of course, translation is basically the synthesis of these polypeptides, which will obviously take the ribosome, and it'll actually take 3 different types of RNA in order to pull this off. So let's take a look at those 3 different types of RNA. Traditionally, back in the day, we used to recognize 3 types of RNA. Messenger RNA, which is essentially the code that determines which amino acid is chosen. Then ribosomal RNA, which is a structural component of the ribosome. And then transfer RNA, which is basically the translator of mRNA code into amino acid. So I oftentimes will refer to transfer RNA as the Rosetta Stone. Right? Anybody know what the Rosetta Stone is? Yep, exactly. Usually I get the program. That's usually the response I get. But yeah, you're right? The Rosetta Stone was significant in archaeology, right? Because it was basically the first time that we were able to actually decipher hieroglyphics, right? Because hieroglyphics were a pictorial-based language, and we had no pictorial-based languages in the world at that time. So we didn't really know what to make of, like, okay, what does the eyeball mean, right? Or what does the cow mean, right? So we didn't know what that meant. We knew that they were words, but we didn't know what they meant. And so the Rosetta Stone has some sort of a description on it. I don't even remember what it was, but it's an era of history where the 3 different kingdoms overlapped. That is to say, when the Romans and the Egyptians were in play, which basically brings it to roughly kind of the late centuries BC, right before it turns over to AD. You're probably talking about maybe the reign of Augustus in and around that area, because you had 3 main inscriptions on it. You had Latin for the Romans, you had Greek for the Hellenists, right? Because that's mostly what was left over in the world after Alexander the Great died. It was mostly a Hellenized Greek world, and Greek was the common language of the world at that particular point. So Greek would be like the common language, universal language. And then you had hieroglyphics reflecting that the inscription was related to some sort of Egyptian stake at home. And of course, the reason why you use hieroglyphics is because that was the traditional ancient language of the Egyptians. The only reason why we'd use that is as a show of goodwill to your conquered, subjugated Egyptian continent, which is what happened throughout the Ptolemies after Alexander the Great. Basically, it was subjugated, but there was even though the Ptolemies were in charge of that area of Egypt and all of North Africa, they still very much were cognizant of the political acuity of maintaining and talking up and appealing to traditional Egyptian culture, because the Ptolemies were Greek in charge of Egyptians. You can imagine what that would be like to have somebody in charge of you who is not your person, right? That would be kind of like Putin coming here and running the US, right? So you would probably, as to quell rebellion and things like that, you would make obvious efforts to say, like, listen, this is your culture is important to me. I'm going to learn hieroglyphics. We're going to write inscriptions in hieroglyphics because that's who you are, and we respect that, and we're all on board with that, right? That's kind of the way it was. That's the reason why you had 3 of those inscriptions. Well, here's the thing. We knew 2 of those. We knew Latin, we knew Greek, and we used those to decode hieroglyphics. The same way, tRNA has 2 different languages on it. It has nucleic acid language and its activity as an RNA. It's a tRNA. But then it also has the ability to translate that into amino acid language. So you're actually going from one language of one molecule to another. And that's the reason why I call it the Rosetta Stone. However, we have also discovered lots of other types of RNA. There's small nuclear ribosomal RNA that's involved in splicing. We'll talk about that later on. There's also signal recognition particles in RNA. This is for protein localization. And then we have microRNA, lots of little chunks of RNA that we used to think were just degraded products. But now we realize they're actually regulatory. They're small, easily overlooked. But now we realize that there's a lot of little bits and pieces of RNA out there, and they're regulatory. They have the ability to regulate different functions and things like that. So our knowledge of what the role of RNA is expanding, and over the last 20 years has expanded like by leaps and bounds. So we've learned a lot about RNA in the last 20 or so years, and that it's not just the top 3. The top 3 were the standard bearers, probably up until about 2000. And then we started adding all these other types of RNA that just kind of started blowing our minds. And we just started to wrap our heads around how functional RNA was, just on its own. So that obviously led us then, right? Because if you've got this code, right, mRNA, that is translating essentially nucleic acid language to amino acid language, you're selecting your amino acids with this code. Obviously, the next obvious question would be, well, what is the genetic code, right? Now, a lot of probably individuals assumed at the time that whoever figured out the code was going to win the Nobel Prize. This particular race wasn't quite as heated, probably for a lot of reasons, is because, well, Crick was involved in some of this genetic code work, and you still weren't that far displaced from the travesty of the DNA structure. So there was probably some pretty strong visceral emotions at this point, which probably tempered that full throttle race to the genetic code. So, Leif, question. Well, first of all, that's up to the Swedes. And I think then there's a series of rules that the Nobel Prize Committee actually sets down. One of them is no posthumous awards. I think the other one is there's no take backs. Well, that's exactly what happened here, and they didn't take it back. So, well, no, Watson and Crick did nothing. They didn't do a single experiment to actually get to the structure of DNA. Now, in Crick's defense, he did do a lot of work on genetic code. But so, I mean, there was kind of a partial redemption for Crick. And that's the reason why I say Crick sometimes is seen as sort of like you just have really poor judgment in friends because you kind of aligned yourself with a real jerk, and we kind of fault you for that. So there was a little bit of that, and so he was able to kind of redeem himself at least partially, not completely, because he actually did put legitimate work into this. He actually did do some legitimate work here. So I'm not going to take that away from him, but clearly this was not Nobel worthy, and we learned our lesson there, right? Because that was a big oopsie moment. I think probably in retrospect. To be honest with you, I would put above Crick my boy Sidney Brenner. Sidney Brenner is the guy who developed C. elegans, which is the organism that we work on in the research lab as a model organism. And he worked a lot on splicing and developing the C. elegans organism back when nobody really knew what a C. elegans was or cared about it. But he set it up and started working with it. And so what they actually found as they started to characterize the code is they noticed that you have these blocks of 3 DNA nucleotides. Basically, they noticed that the code is read in 3-letter words. And that was important, right? Because now what you do is you have your code organized into 3-letter words. Well, how did they know this? Well, what they did was they produced frame shift mutations by either adding single or double insertions or deletions. That is saying one or two nucleotides into an already established frame, right? So here's what a frame shift is. Here's what they noticed. I'm going to write down a piece of DNA here. So what they noticed was that when you read the amino acid assignments, that it was read as 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, so forth. How do they know that? Well, let's go ahead and add one. Okay. So if you add a nucleotide to the mix, then instead of basically shifting, what you have is now this becomes 1, this is 2, and this is 3. So that's your new word right there. This is 4, 5, and 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, and then 16. Notice what's happened. You've shifted everything in one direction, right? So now all of a sudden, instead of your 1, 2, 3 is AAA, but notice your 4, 5, and 6 is now ACC instead of CCA. So what's happened is it's kind of turned it into nonsense. It's just gobbledygook now. Now this is with an insertion. And this happened with 1 or 2, but not with 3. Now let's go ahead and do a deletion. In a deletion, what I'm going to do is I'm going to take away this one and this one. So now this one becomes 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14. Now notice what happens. Now all of a sudden, instead of AAA, your first one, it's now AAC. So everything is shifted in this direction. That's if you have a deletion of 1 or 2 base pairs. So you shifted your frame. What happened when you did 3? Well, as it turns out, it wasn't as bad. So here's an example of a frame shift, right? So here is basically the area that I'm going to look at. Notice it's normally ACG. I'm going to take out, in this case, C. So it's going to go AGC. So this is your new codon now. So now notice everything else is just nonsense here, right? So it's like instead of histidine, arginine, asparagine, it's now threonine, alanine, threonine, histidine. It's like, what? It's just like you're just gibberish, right? It's like protein gibberish. So this is a frame shift. Happens with either 1 or 2 deletions or insertions, either one, right? This is a non-frame shift. Why? Because what happens is you use 3. Let's see what happens there. So here's your ACG. We're going to take that entire thing out right there. So it's going to go from CCU to CAC, CCU to CAC. What happened? What happened was we omitted threonine. But the rest is normal. Histidine, histidine, arginine, arginine, asparagine, asparagine, alanine, alanine, serine, serine. The rest of it's normal. All you did with 3 is omit that one selection. That tells you, along with the frame shift, that you're reading this in 3s. So the other thing they wanted to know, once they now realized that it was being read in 3s, is they wanted to know whether or not whether the spaces counted, right? So do the spaces count? That's what they want to know. And so here's how it looks, right? So in this particular case, if you have this situation, this is where the spaces count. So what we'd have, say for instance, if we took a sentence with just 3 letters in it, with spaces between them. So what we do is if we deleted T, the question is, does that space count? So if we took this one like this, and we omitted this one, does this count? So is this still 1, 2, 3? Right? Is 1 now empty, but it's still counted? Or does it shift over like that? What we saw was actually the spaces don't count. So in this case, if we omit the T, what we see is everything just shifts over and we just start counting for the next one in line. So this is basically what we found, was that basically that the spaces don't count. Okay, so that's important. That makes a lot of sense. But here's the thing. So we know there's codons now, right? We know that the codons somehow assign your amino acid. But here's the question. Which codons select for which amino acids? Well, Marshall Nuremberg did a lot of this work. And it was very simple. Basically, what Nuremberg did was essentially create an in vitro translation assay, like just basically like a cell extract with all the translational proteins in there. So everything was there that you needed, right? All you needed was an mRNA for it to work off of. That Nuremberg made himself. So he'd make his own stretch. So for instance, he'd say, okay, let's go ahead and do this. Let's give you a stretch of U's. Feed it to the translation mixture and then collect the protein that it makes after that and thus identify that. What he saw was this. Two things, first of all, that really nails things down. How many phenylalanines do you have? 3, right? How many nucleotides did you feed it? 9 divided by 3 is what? So you have 3-letter words. The only way you could get 3 phenylalanines from 9 U's is if you were reading these in 3-letter batches. This confirms the work that was done previously by Crick and Brenner on the frame shifts. It also basically said quite substantially that if you have UUU, that is the designation for phenylalanine. Now you can imagine this would be a very simple experiment now that you got to set it set, right? Let's just start changing it up. Let's feed it some UUA, some UUC, some CCC, some AAA, some ACC, some ACA. I mean, let's feed it whatever we can find it. And eventually you collect all these assignments. And then you can get basically where all these codons are assigned to specific amino acids. He also noticed a couple of other things. He also noticed 3 different codons, which were stop codons. So U, UA, UGA, go Bulldogs, and UAG, right? Sorry, I just slide that one in there. Right, so these were stop codons. So how do you know that? Well, because if he did this, what would he get? You get a phenylalanine, right? Another one, and then nothing, right? Because UUA is a stop codon. So that's how he figured out the stop codon. He also noticed that there was a start codon, an AUG, which encoded a methionine, and that every protein actually has a start codon. The very first amino acid it has in line is a methionine. So over time, you can collect all of the different assignments and you can organize them into a big table. The other thing he also noticed was the code is degenerate. That is to say that some of these amino acids will have multiple codons that encode them. So then once you organize all this data, you come up with this. Wow, what is this? This is genetic code, right? Basically what you do is you read your first letter of your codon down. The second, you're going to read across. And the third, you're going to read down here. So here's a good example. ACU. What is that? Well, you're going to read down first. So A, so this is the row that you're looking at, right? Then you're looking for C. So you go to this column here. So you're looking for this confluence. So here's AC. And you're looking for U. So the U is going to be right there. What is it? Threonine. So one of the best things you can do to practice this, and I did this in my other section, is take a piece of DNA template. Like that. And then do a replication. Make the other strand, right? The other DNA strand would be TTG, TTT, GCA. Yes? Now what you want to do is you want to do transcription, right? What's complementary to 5'? 3', right? And then what do I have? U, G, right? U, G, C, A, right? 5'. Now notice it's inverted, isn't it? Important thing. This is all 5' to 3'. Everything here. So notice how it's flipped around. before you analyze it, you want to flip this around. So what you would actually translate is 5 prime A. What, that's a ACG. Sorry, that's a C. Right? UUU GUU 3 prime. That's what you would translate. This would be your mRNA. By the way, by convention, whenever you see sequence, it's always written 5 prime to 3 prime going left to right. So whenever you report sequence, like we just did here, you always have to flip it around, okay, before you analyze it. So what would this be then? If we translate it, ACG is what? It's a threonine, right? There's ACG right there. What's UUU? You know that one. That's phenylalanine, right? So basically, if you take a look at UUU, there it is right there. That's a phenylalanine. Last one, GUU, right? In this case, you're coming down here. There's your GUU. It's a valine. So notice what I did. The best thing you can do with this is practice this. Just write any random DNA sequence as big as you want, replicate it, then do a transcription of it, then do a translation of that transcription using the genetic code table. That'll really help drive home the basic flow of how this process works. And then what we can do is we can take a look at our cute little pigs. And we'll do that next time.